also recording the first half if you need to go through anything again. Okay, so now let's look at something called codominance. The types of questions that we covered so far is incomplete dominance. Again, incomplete dominance is when you have the blending of alleles to form a third allele. You tend to see that in flowers, like the petal colors. You tend to see pink, which is formed from red and white. You also saw that question with the mouse, with the gray color, which was formed from black and white. That is incomplete dominance. Then you had complete dominance, which was the questions we did with tongue rolling, hair color, eye color, where you have something that can, if you have a dominant allele, it can overshadow or express itself if it's in the heterozygous form and the recessive one is silent. Codominance is different. Codominance means together. So you could have a case where you have both types of alleles, but they are both dominant. So you have two that are dominant and they are being expressed together. You tend to see this kind of case in blood groups and a genetic disease called sickle cell anemia. So in this case, you're going to have only capital letters. All right, so let's look at the first. So, all right, so codominance is sometimes neither allele dominates the other. All right, so no one is stronger than the other, such that the influence of both alleles is visible in the heterozygous individual. So only when you have the two different alleles, that's heterozygous, that is where you see codominance taking place, which suggests that both of the alleles are being expressed at the same time. So this example here on the screen is in blood groups. You have three different alleles for blood group. You have a capital A. Now this could be expressed in two ways. You could have two big A's or you could have A, O. Blood group O is what you call a recessive blood group, right? It's not very common. Whereas blood group A and B tend to be the dominant forms. Now there's a special case where you are seeing this highlighted one in red. All right, so first let's go through all of the blood types. Now this is something you all should know. Normally when you do a blood test, they're not going to tell you the alleles. They will just tell you if you have blood group A, B, or what have you. All right, but let's go through. So first you have AA, which is blood group A, AO, which is also blood group A. So in this case, A stands out. O is recessive. If you have blood group B, we understand that's blood group B. If you have BO, it's understood as B because O is silent in this case. Now this is the special and unique one. You rarely have people with this type of blood, by the way. This is blood group AB. So this means that person have both types of blood groups. They have both blood group A and they also have blood group B being expressed. So this is the codominant blood group. Right? Do you all know anyone who has blood group AB? Right? So very, very rare. You don't really meet people with this type of blood. Now, do you all know your blood types? That's something I should ask.
I come in. I'll bring it.
Miss, my father is AB.
Oh.
Okay, well, that's very special. And I'll tell you why. So, someone's mic. Alyssa's mic. Yes. Now let's look at a question here. Here we are seeing something. This part cut off. This part not looking now. All right, let me just fix something. I noticed a little error. All right, I'll use a different diagram because this one looking a bit mixed up. Let's say we were crossing blood group A with someone who has blood group B. So let's say one parent have blood group A, one have blood group B, but they also carry the recessive allele, which is blood group O. All right, so let's multiply that and see. And right, I always need a whiteboard for this type of question. Now I want you all to cross it in your own, at least do it in do it in your book and you could follow along. Right, so here we have, let's cross right? Let's cross blood group A with blood group B. You'll go ahead and cross that. But I want you all to use the combination where there's a recessive allele. So in that case, your parental genotype should be AO crossing with BO. Right? Cross that and tell me what you got. Right, so here we have AO. BO. Right, so A crossing with B. So you get blood group AB. A crossing with O. B crossing with O. Don't ever put O in front. So in the event you're crossing and you see you make an error and you put 0. Sorry. You put 0. B, make sure and always put the dominant forms first. So you put B, O. I've seen some people put in O, B. Don't do that. Now look at this. You have O by O, and you get two O's, right? Which one is codominant here? Where both blood groups are being expressed at the same time.
AB, miss.
AB. So let's put a ratio now. So here we have, right? So I'll put a second sticky note here. So F1 generation genotype will be AB, which is codominant, BO, AO, OO. In terms of the phenotype now, F1 generation phenotype. You just have to put, it's a lot of writing, so you just put in the word blood group in front. So blood group AB, right? And just list out all of them. It's a bit. Right. Blood group. I'll put BG for short. BO. Actually, no, that'll confuse people. Blood group BO. Blood group AO. Blood group OO. And then you put a ratio at the end. So this one is a 1 to 1 to 1 to 1 ratio. A lot of possibilities in this case. Right, much better. Right, I want to post this on my slide. Let me take a little screenshot there. Bring it across. Right, so this is just an example. Now let's look at another one. This is as if blood group O, I know this first line is a bit funny. So this is as if you had a parent who is recessive and you have a codominant parent. What is the chances? So here we are seeing one chance the child can have blood group A, or two chances the child could have blood group A, two chances the child could have blood group B. All right. So now you all have to know how to put your, some people just make an error and put capital and common and codominant. Codominant, anytime you see a question with blood groups or sickle cell anemia, which we are coming to, only use capital letters. The minute you put a common letter in this question, this type of question, you're going to get it wrong. All right, so let's look at another one. Sickle cell anemia now. Right, I'll fix this diagram. Now let's say, now this disease is a genetic disease where your red blood cells tend to have an abnormal shape. You call it sickle cell. It's shaped like a crescent. Normally, your red blood cells tend to be a biconcave disc shape. Someone who has sickle cell anemia, which is a very rare, you would see the red blood cells are in this kind of deformed shape. And that is going to pose a lot of problems. Now, they could live a healthy life, they'll have a normal, what will happen is they will feel weak. They tend to feel weak because less oxygen is being delivered, right? Whereas if they had only healthy red blood cells, they won't have that weak kind of feeling. So there's no way to cure this disease. There's no way to get rid of it. They just have to live with this particular disease. So here we are seeing the person have both kinds of red blood cells. They have normal red blood cells and they have few in between sickle cells. So some people tend to have codominant case where they have some that are normal, some that are abnormal. So in this particular question, you tend to use capital A for normal. They call it normal hemoglobin. Hemoglobin is just the iron or the protein in your red blood cell that gives it its unique shape. So normal hemoglobin, we use capital A. If the person has a sickle cell allele, we use capital S. So abnormal hemoglobin, you use capital S. The normal allele, HbA. Now, Hb stands for hemoglobin. So some questions, they tend to put hemoglobin just to represent where the allele is found, right? But you don't have to put this when you are working it out, all right? So here we are saying normal A or normal hemoglobin stimulates the production for normal hemoglobin. The abnormal allele, which is HbS, hemoglobin S, stimulates the production of abnormal hemoglobin S. So let's cross some parents to see. I don't like this, how this chart looking. I'll re-upload the slides with this new diagram. I find this looking a bit difficult. Okay, so let's say we have, now you will have a question to do on your own, so follow. Let's say we have the parental phenotype as someone who is normal and someone who is codominant. Now, if someone has sickle cell anemia in the codominant case, we use the word carrier. It means that they're still healthy, they're still normal, but they carry that sickle cell trait, right? So let's put parental genotype now. Now, some cases, people tend, in some questions, they may put the hemoglobin symbol, and then you have to put the allele, but let's make it simple. So capital A is normal, whereas the carrier will be capital A, capital S, because S is for sickle cell, right? So let's just show using a Punnett square now. So we have A crossing with ES, which is sickles, the codominant case. So here we have two big A. Here we have a codominant case. Here we have a normal. The person is normal. Here we have codominant again. So this ratio is a 1-to-1 ratio. So you put down your parental phenotype. Sorry, F1. F1 generation genotype. So we have AA, AS, AS. Then you put the phenotype. So you have AA is normal. And then you have carrier. You call it carrier when they have the trait, or when they have the trait and they also have normal red blood cells. This ratio here will be a 1-to-1. Why? Because it's 1, so it's 2% or 2 chances they're normal, 2 chances the child being a carrier. And when you narrow it down, it is simplified as a 1-to-1. Right, just now we come into our question. Right, so I'll put this now in the slides. So that's an example. Now for these questions, always use these symbols. They tend to give you it, or always use, because it's understood as A will be normal, S is sickle cell, right? Okay. Right, so this is the shape of normal red blood cell. This is the crescent shape or sickle cell. Let's look at sex inheritance. Sex inheritance is to determine the sex, the biological sex of a baby, whether it be male or female. So this working will be standard. This is a 1 working if ever you have a question with this. So in each cell, 1 pair of chromosome is composed of sex chromosomes. You could either have 2 X and 2 X and a Y. So for females, it's represented as XX. You always use this symbol for female. The genotype for male is always represented as XY. Now, the male is the one who can pass on either the Y chromosome or the X chromosome. So it's consequently, the father is the one who is actually determining the sex of his offspring, because the female already have 2 X's. So it's up to the male now to pass on either the X or to pass on either the Y. So that is why they said males are the one who determine the sex of the baby. So let's look at the working. You put down the phenotype, so it's female mating with male. You put down the genotype, XX crossing with XY. You circle your gametes. Now this is a test cross, so here we have 50% chance it being female, 50% chance it being male. So anytime you have to determine the sex of a baby or offspring, it's always 50-50. All right, so in any question that you have to show the working for the inheritance of the sex, you always use these symbols. This one is very straightforward. Right. This is something I want you all to do for homework. What is meant by each of the following genetic terms? Sex-linked genes, heterozygous. You all already know these two. So I want you all to come up with a definition for sex-linked genes. And that will be the last type of working we have to learn. So let's go through sex-linked diseases. Now, sickle cell anemia is not a sex-linked disease. This is a regular disease, a regular genetic disease. Sex-linked diseases is different. This is when you have a disease that is, the allele is found on the X chromosome. So since we already look at the sex chromosomes here, we have to know what is a sex-linked disease. The only two that you have to learn in your syllabus will be hemophilia and color blindness. Now, why is it that the allele is found only on the X? Simply because the X is longer than the Y. There's more space for a genetic disease to be attached. All right, so what exactly is hemophilia? It's a sex-linked condition where the blood fails to clot at a cut. So if someone was to get a cut, they will bleed profusely. It's going to take very, very long for a blood clot to form, or it might not form at all. And this is very rare. These individuals cannot get surgery. If they get a wound, they can lose a lot of blood. So it's very, very rare type of disease. Okay, so here are the different genotypes. Anytime you're doing this particular question, you always have to put your sex chromosomes. All right, so for female, the female could be, now we use capital H for normal. That represents normal. We use common H here to represent hemophilia disease or hemophiliac. So if you have this combination, the female is normal. If you have capital H, common H, the female is a carrier. So what does this mean? It means that she's not suffering from the disease, but she's carrying the recessive allele. All right, so she's still normal. She can also be a hemophiliac, where she have both alleles as hemophilia. Males are very narrow. It's either they have the disease or they don't. All right, and that's because they only have one X chromosome. Nothing is attached to the Y. So here we have the normal hemophilia allele. So he's normal. And then we have the recessive allele or the hemophilia allele. So therefore he has the disease. So their chances of getting the disease is way greater than a female. All right, so here is a little example showing someone who is normal and someone who is a carrier. The father being normal and the mother is a carrier. So we are seeing that if you were to have parents who are normal and a carrier, there's a 1 out of 4 chances, so a 25% chance, the male child can be a hemophiliac. All right, so there's a 25% chance the child can be a hemophiliac. Whereas there's a chance, there's 50% chance the child can be normal. And there's also a 25% chance the child can be a carrier. All right, so I want you all to go through and look at this example. There's one more. Now, before I go into color blindness, there's one more. There's a question pertaining to hemophilia I want you all to do in the worksheet. So let me carry you back to that worksheet. Here it is, but this one here. Now, these questions was cut out from past papers, so you would see C, and then it's, all right, so it's this second C here. Right, Jaden, go ahead and read part C.
By means of a genetic diagram, show the genotype and phenotype
No,
of the
this
person.
first part, the first
Okay.
part.
The genotype of Mr. Henry is XY, and the genotype of Mrs. Henry is XE, is XX. X and Y represent the sex chromosomes. H represents the dominant normal allele, and common H represents the recessive allele for hemophilia. One of their two children, John and Jenny, inherited the sex-linked disorder, hemophilia. Using a clearly labeled diagram, show how the hemophilia disorder is passed from the parents to one of the children. Your diagram must include the genotype and phenotype of Mr. and Mrs. Henry, and the children. The beginning of the diagram has been done for you as follows.
Okay, so this type of question, you're only going to use these symbols. So don't come up with your own alleles. You have to use what the question gave. All right, so let's work this one out. Right, they already did part of it. Right, so here we are seeing the carrier female mating with a normal male. The parents is X. Well, the female, now you have to look, I know you can't really make out the letters. Here we have a carrier female, so you have capital H, common H. The male, you have capital H. This is a minus sign. That minus means that there's no allele. There's never an allele present on the Y chromosome. Right, so just put a minus sign. So let's do the cross. So we have X. Capital H. Then we have X. Now be very careful with these because you have that chromosome as well. It can look a little bit confusing. Right, so it doesn't matter if you put the female and the male on the top or the side, does not matter. Right, so the male, he is normal. Y, there's nothing on the Y, right? So when you're multiplying this, I suggest when you're crossing this, multiply and put down your chromosomes first. So put X, X. Then put in the alleles now. Right, so let's put it. So we have H, capital H. So we have capital H, capital H. Right, we're going again. X, Y. So you put down your X, you put down your Y. Then you go to your allele now. In that way, you're not going to confuse yourself. Right, then you multiply your chromosome again, X, X. Right, I'll just put it down, X, Y. So now you just have to fill in the alleles. So capital H by common H. So you put your capital first, then you put your common. Then you do it again. Common in this case, now you don't ever put an allele on the Y. You put it on the X. So this is the working. So here we are seeing you have a normal female here. You have a carrier female. You have a normal male and you have a hemophiliac male. Same thing like the example on the slide. The same working. Everyone seeing that?
Yes, miss. Yes, miss.
So for homework, I want you all to do the remaining questions. It's just this one here, right? They give you the symbols. There's also one on color blindness right here, part B. What is color blindness? By means of a genetic diagram. Color blindness is color blindness is also a sex-linked disease. So when you are showing you're working for color blindness, you also have to put down your sex chromosomes. Now, someone who is colorblind simply means it's also known as red-green color blindness. So this is the person, they are unable to distinguish between certain colors. All right, and these alleles are only carried on the X. Same way as the hemophilia disease. So in this case, you could use C for normal, common C for colorblind when you are working out your question. And don't forget, put down your sex chromosomes first. So follow this example. Next week, I want to correct, and I want to see your workings. Some people posting working, some people not. You are doing part B and C, and you are doing the question 5 on page 7. Right, I want to see it working. I want to, I'll show you on screen, and then we have genetic engineering. We have completed this entire section of the syllabus. All right, so are there any questions?
No worries.
All right, so
No,
I'll
miss.
leave you at this point, finish off those questions, and next week we'll return to correct. Have a, oh, and you all are back to school from next week, so I wish you all a good first week back, and I know that, you know, your minds are already in exam mode. You have 2 weeks back at school, and then you will be at home studying. All right, so have a good weekend. Take care.
Bye, miss.
Bye, miss.